A very good evening to everyone. Am I audible to all delegates?
Audible, sir.
Thank you. Thank you so much. We're just waiting for the resource person to join. So again, a warm welcome and very good evening to all dignitaries, organizers, esteemed faculty members, and all delegates presented in today's online session of this 3-month certificate program on research methodology and biostatistics. Today, uh, it is with great pleasure that we introduce the resource person for today's live session, Dr. Prashant Bedarkar, sir, currently serving as a professor CAS at the Institute of Teaching and Research in Ayurveda, ITRA Jamnagar. Sir brings an impressive 17 years of teaching and professional experience with over 13 years dedicated to postgraduate teaching and 12 years as postgraduate guide. He has also guided PhD students for over 5 years. Dr. Prashant sir has made significant contribution to the field of medical research with 122 research publications in peer-reviewed and index journals, amassing over 30 citations. His work includes 9 single authors publications, 2 as a main first and corresponding author, and 111 as a co-author. His research articles have been indexed in notable databases such as 30 in PubMed NIH, 10 in Scopus, 20 in PubMed and Scopus, and 21 in Ayush Research Portal Dhara. Publication of 26 original research works. His research endeavors has focused on various areas including pharmaceutical analytical standardization, pharmacological studies, and clinical research. Dr. Prashant sir has been principal investigator for significant research projects funded over 10 lakhs INR, including an ongoing projects on the pharmaceutical standardization of Abraka Bhasma and Nagarjuna Rasa with the pharmacological evaluation of metabolic syndrome and cardioprotective activity funded by IPGT NRA. Also, as co-investigator of the project completed on systematic review of research on Tamka Swasa, bronchial asthma in 2012, funded over 9 lakhs by IPGT NRA, and also on SOP for standardization of identified Ayurvedic formulation, herbo-mineral preparation, visualized Rasa, Manikya, and Tribhuvan Kirti Rasa in 2017, funded over 5 lakhs by CCRAS. In addition to his research, Sir has contributed to academic literature by co-authoring books and monographs on topics like the safety and efficacy of metallic herbo-mineral Ayurvedic formulations and monograph on Raskar in 2015 and studies on Naga Bhasma in 2023. He has been an active participant in conferences and workshops, delivering invited guest lectures and presenting his research at both national and international levels. Contributed as expert for policy document Ayurvedic Standard Treatment Guidelines, first edition 2017, Ministry of Ayush, Government of India. Dr. Prashant sir expertise extend to his role as an associate editor of the international journal Ayu and his involvement in developing e-content module for Ayurveda pharmacists. He has also contributed as an external editor for several journals and served in various institutional roles, including as an evaluator and examiner for national and state level board and universities. Sir has made significant contribution to the field of Ayurveda through his extensive supervision guidance of thesis work, having awarded the degrees of 46 MD, MS, MPharma, and PhD candidates. Dr. Bedarkar has also been a co-investigator on a patent application for a novel drug for CCRAS CoV2, published on 28/5/2021 and currently under examination. He has presented 8 papers and seminars and conferences and workshops. He holds membership in scientific organizations, including the Society of Pharmacovigilance India and the Board of Studies at Dr. D.Y. Patil School of Ayurveda, Nehru, Navi Mumbai. Among his accolades, he received the Rasa Vibhuddha Award on National Ayurveda Day, 2nd November 2021, from Dr. Vasisth Ayu Remedies, a national recognition. His other co-curricular activities include serving as external editor of journals such as BLD's University Journal, GREIM, Ayu Home Journal, and Shillong. He has also played various institutional and university roles, including physician, extension OPD at Central Jail, Jamnagar, and multiple in-charge positions at Gujarat Ayurveda University. Today, we are honored to have Dr. Prashant sir with us to share his extensive knowledge and insights on this live session. Please join us in extending a warm welcome to sir to lead today's session. Please, sir, welcome, sir. Please unmute, sir, at your end.
Yeah, Amidji, am I audible now?
Yes, yes, audible, sir, audible,
Okay.
sir.
Thank you. There was problem with sharing, and I was not able to make me unmute. Okay.
No, it is
The
visible,
PPT is visible.
visible, sir, on
Okay,
third
okay.
slide. We are on
Okay,
third
okay.
slide.
Okay. Thank you. So, Amit ji, very much thank you. I couldn't get to recognize that I was muted. I was urging you to let's have a brief. It was very huge introduction of mine, and I'm very awkward to listen to myself. बहुत ही काफी ज़्यादा कुछ बोल दिया आपने, ऐसा कुछ है ही नहीं. Thank you for brief information. So today's topic given to me is traditional drug discovery process and latest trends in drug development. We shall discuss it in two parts: traditional drug discovery as well as latest trends. But sometimes we may go hand in hand, and we shall focus more on not evolution of newer molecules or contemporary discoveries, but what can be derived as a new drug from traditional medications as a novel treatment, as a new molecules or a treatment, whatever it is. What is the scope? We shall be very much restricted to that as per my expertise is concerned. I'm not an expertise in modern pharmacology or toxicologist. Likewise, as per as my opinions are there, I shall share some things in this regard. So, drug discovery in traditional medicine what shall be covered and under this heading, traditional drug discovery. So all drug discoveries in traditional medicine and traditionally used drugs, their dosage form modifications or formulations, and authentication of drugs, involvement of newer drugs or possible variants of these drugs. And drug discovery as per traditional methods means Ayurvedic aspects of drug discovery we have to take into consideration. Research on application of principles of traditional medicines or Ayurveda medicines like Yogvai property, their adjuvants, Pathya, mode of administration, effects in people with different clinical variants like Prakriti, Agni, Kost, Bal, Vadhi, Avastha, etc. In a variety of clinical setups, their applications and research in this regard. So, literary research related to drug, that is review of type, nature of drug, likewise, and preclinical, experimental, or clinical research is also incorporated in traditional drug discovery. So, we shall see a few scope or role of latest trends in drug discovery, which can be incorporated into exploration of traditional drug discovery and can evolve as a potent new innovations or ideas or pharmaceutical applications or therapeutic protocols or remedies. So traditional drug discovery was what there in situ in classical context or classical era. So it is very much clear that every aspect which we encounter with drug discovery, that was there in the classics, say literary research, synonyms of drug, commentaries, therapeutic parts of parts mentioned in ancient classics, classification of drugs, systematic systems of measurement, classical formularies like pharmacopoeias. All those suggest that there was literary research as well, and research on application or principles of traditional medicines like Yogvai property, adjuvants, Pathya, mode of administration. All those suggest that timely it was evolved. Timely adjuvants were evolved. Mode of administration was changed. Uses of drug formulations that was changed from ancient era till the date, and it suggests that there was stronger and stronger, more and more researches even on principles. And current researches on this principle suggest its potential, like yogai property of say madhu, shilajatu in the form of formic acid,
Sorry
fulvic
to
acid,
interrupt you,
likewise.
sir.
Yeah, yeah.
So slides are not in slideshow mode and not changing, I think. We received
Now slide has changed now.
not changing, sir. We are on first slide
Oh,
till
slide has
Right?
on third slide.
uh, second one now.
Now second, but it's
Third.
not in slideshow mode, sir.
Yeah, yeah. हाँ नहीं हो रहा। अभी हो रहा है, चेंज हो रही है स्लाइड्स।
We are on third slide, sir.
अभी चेंज हुई?
No, sir. No, sir.
या अभी स्लाइड चेंज हो रही है?
On second slide. On yes,
हाँ हाँ,
it's changing.
लेकिन
But
मुझे लगता है वो स्लाइड शो मोड में जाएंगे तो नहीं होगा। एक बार करके
okay.
देखता हूँ अगर हो पाया तो। अभी चेंज हो रही है स्लाइड्स?
No, sir. No,
नहीं
sir.
नहीं हो रही है ना, तो नॉर्मल मोड में ही अगर करेंगे तो अच्छा है। मेरे एंड
Okay.
से कुछ
Okay, sir.
प्रॉब्लम्स
Okay.
है।
Okay, sir. Okay, sir. All right, sir. Please continue. All delegates, I will share the slide after the session with all delegates, so you can go through it.
So there are
Sir.
researches in Yogvai property like Gomutra distillate of cow urine, Shilajatu, and its contents like fulvic acid and normally fulvic acid and humic acid, likewise. Even on Tricot2 piperine, there are patents got in this sector, so which suggests the potential of even principles like in adjuvants Again, there are many patents on mode of administration, adjuvants. For an example, even on Gomutra distillate as adjuvant in antibiotics, antitubercular, anti-cancer drugs, likewise. And for adjuvant also, drug-drug interactions, many patents are there. So pathya, apathya, these are potent areas for exploration in traditional drug discovery. So classical drug research, it is addition of single drugs, formulations, timely, those are many drugs are added to pharmacopoeia, preparation of formulary, pharmacopoeias like Nigantu, etc. Varieties of single drug, their acceptable varieties or features, Grahya Grahayatva, best varieties, likewise. Advancement in pharmaceutics like operations, instrumentations, Shodhan, Zaran, Maran, Amrutikaran, Lohitikaran, these were timely, those are evolved. And even some principles like Amrutikaran, the patents for fatty acid complexes of iron, there are patents. The process is just similar to Amrutikaran, Bharjan Greek, Gairik Shodhan, Amrutikaran of Loha and Abrak, likewise. So there is immense potential. And recently, I may not be there in slides, but recent 2008 patents on copper 2 ions and iron 2 ions in the form of ferrous, their tannates and antioxidant organic groups. They have been patented for extension of extended antimicrobial activity, extended antioxidant activity with these ligands, organo-inorganic ligands. So the word is coming back to organo-inorganic ligands, which is a part of Rasa Shastra. Due to scarcity of phytochemicals and plant origin drugs and plant derived drugs, the word is again regaining the shapes, and there are patents, and those are recent patents. We know that you used to give bhavna, anupan, and many operations are done with kasis and tutha, with nimbuswaras and tannin containing drugs, and tannin complexes of tannin with this iron sulfate and copper sulfate in the form of +2 ions are taken as a patent for these activities. And nothing has been spared, none of the antioxidant molecule. And for tannin, they have taken extracts of 3 tannin containing rich drugs. So currently, we are people are exploiting, either get explored or people will exploit you. They will take the benefit of your principles and pharmaceutical operations and drugs, and we shall lag behind. So the current trends of drug discovery, until and unless we adopt, we will be lag behind in sense of advancement of drug discoveries. So there was in silico methods application, yatha lohe tatha dehe. So Ranjan, Kraman, Vedakatva, all this suggests that mercury should have specific reactivity with different metals. There are color changes. That means it is assurance of reactivity with certain reactivity of mercury with certain metals. So all these Ranjan Yog, Kraman Yog is associated with the bioavailability and excretion of mercury. So it is drug-drug interaction. So it is in silico methods or molecular docking and targeted drug delivery. For targeted drug delivery, again there are patents with silicons, nanosilicons, porous silicons like kaolinite, khatika, we used to use for parat bandhan, shodhan in many activities. Abrak basma as a yogvai, so liposomes as a drug delivery system. So recently there are multiple patents, and people are exploiting, taking benefit of all these principles which we do have in silico methods, specific reactivity towards specific molecule, inorganic molecules inside the body. So it is very much sure that there was systematic drug development, and even in silico methods and targeted drug delivery, everything was there in situ. Right now we used to use encapsulation, micro encapsulation. We used to give bhavna with albumin and proteins, the bhavna we used to give. So we used to use gugulu kalpa, where the gugulu content is almost 50%, and this is encapsulation of resins which are which is targeted drug delivery, being insoluble, and the efficacy increases several folds, because the dose of these formulations is say 1 to 3 grams, and the rest of the powders are there 50%. So those are subtherapeutic doses of these powders, combination of powders, say 20, 15 in a variety of Bubulu preparations. Those are subtherapeutic, and we don't expect any therapeutic activity with this. This activity enhancement is due to targeted drug delivery and various processes of racemization, likewise various processes on these reagents, likewise. So the potential of this racemization, drug delivery, targeted drug delivery, all those we used to incorporate into therapeutics, and there are principles for that, Mardhanam, Gunavardhanam. Now people are working on mechanochemistry with only mechanical trituration and all the chemical changes, size changes in size and shape, likewise. We do have immense potential with Ayaskruti Bhasmas, drug delivery in the forms of liposomes, and protein bounding in the form of Chirpak. Likewise, these are very potent areas of drug discovery, where people are actively involving it, and evolving into pharmacoeconomics of this. Yeah, and even patents. So even there are history of drug development. It suggests that timely Kshatrikaran and different there was a improvement in timely improvement in protocol and guidelines of therapeutic administration, like subsequent administration of Pathya, Apatya, Kshatrikaran protocol, Rasa Loha, and Rasa Abhrak Yog Sanyog. Likewise, these are value addition to which suggests that there was continuous improvement in protocols, which is end outcome of drug research or drug discovery. So properties of Parat for therapeutic use, Kraman Yog, likewise Vikar Shanti Upay. These are Vikar Shanti Upay. It has immense potential as a chelation therapy, and which is although mercury is outdated, but still it is in use. And arsenicals lates are very common even for pesticide residues and many other concerns, organo-inorganical pesticides. We do have immense potential in the form of Vikar Shanti Upay as a chelation therapy. Even Pathya as a bioaugmentators and adjuvants, value addition treatment. So Visha for Kraman, Vedhans, those are also have target specificity, delivery, fast action, assurance of potent effect. This were value add in the sequence of drug discovery in classics. There were initially Ayaskrutis were developed, and then in case of, say, if we take example of Loha, initially in the form of Ayaskruti it was used, then Trivit Pak, Bhasma, Amrutikaran, Dhanyabrikaran, Amrutikaran, likewise several repetition of put up to Shataput and Sahasraput, Lohitikaran. These are value added value addition to pharmaceutical operations, therapeutic recommendations, likewise. And we have discussed this replacement of Yeshad with Rasak, likewise uses of Gugulu, Shilajatu, etc. So addition of new dosage forms, this was also there in the form of dosage forms of Loha Mandur. There are more than 50 dosage forms of Loha and Mandur. I'm not talking about formulation, but the different dosage forms. So Kshira Mandur, Takra Mandur, likewise various pharmaceutical operating procedures leading to variety of dosage forms. So Pishti, Dhruti, Arka, Masi, Bhasma, all those were subsequently derived. So researches on quality of raw drugs starting currently. What we are doing, we do have for research medicinal plant databases like AFI, API. For drug research, Anupta Dravya, we are working on drug development. Government institutions like CCRAS, IMPCL, NGOs, several people are working in this area. And good collection practices, we do have good storage practices and good processing practices. In the form of this, we have ample of data on this processing for good storage practices. Vallatak with effect of duration, there are publications and researches. So on all this on Shodhan, the projects are submitted by CCRAS, and several publications are there in public domain. So importance of manufacturing processes and dosage form in safety, efficacy, and quality assurance of drug and Ayurvedic dosage form. For an example, effect of Nirvapan in safety evaluation, Tamra Basma, it is evaluated twice, and one which was immersed in quenched in water, Nirvapan was done in water, or Nirvapan was not done at all, and Maran was done. It was found toxic as compared to one where the classical methodology of Nirvapan was adopted. So this safety study was repeated twice, and with same it was proven that this process of Shodhan has immense potential. And not only this, and all Vishavarga Dravya, all toxicity studies are carried out, which suggest comparatively more safety as well as efficacy. So many research works are there on importance of manufacturing processes in safety carried out at IPGT and RA. I will not go into depth of that, but with enhancement of put, there is enhancement in safety, like 10 putti abrak basma, 7 putti, 10 putti and 7 putti nag basma is found to be toxic, whereas 30 and 60 putti nag basma there was no toxicity. Even more than 100 times of acceptable levels for threshold levels for toxicity, 10 ppb in blood, lead levels were there, but there was no toxicity in tissue or biochemical parameters. More than 21 organs were studied, and biochemical parameters, neurological behavior, and all physiological behaviors, no toxicity was found. So the same was there with Jaipal, Ashodit Jaipal. There was gastric perforations, ulcerations. Likewise with Shodit, it was not there. And in case of all this, with Vatsanab also, more than 10 times doses of Shuddha Vatsanab and Tribhuvan Kirtiras prepared from this. Even those rats which were having duplex, quadruplex arrhythmias, severe arrhythmias, bradyarrhythmias up to 50 heart beats per minute heart rate. Likewise with fetal arrhythmias, and there was no toxicity in case of this, whereas vice versa, in Ashodit there was toxicity. So a lot of works have been done. We will not go into depth of this. In manufacturing processes also, effect of Mardan, effect of Shodhan, repetitive operations, addition of mercury, without addition of mercury. We have evaluated a lot that addition of mercury value adds to even to safety also. Arogyavardhini Ras prepared with mercury is comparatively more hepatoprotective than without mercury. Addition of mercury in Amvatari Ras has better therapeutic efficacy in relieving rheumatoid arthritis than without. With addition of mercury Swaskutarras and without Swaskutarras. Likewise, many operations are there, and many researches are conducted even on Sanskara, Ashta Sanskarit, value addition of only Anuvasit Sanskar of Parad. The Rasparpati prepared from Anuvasit Parad as that of Ashta Sanskarit. Only one value add Sanskar has improved recovery of gastric ulcers in rats, intestinal ulcerations in rats, and comparatively more decrement in gastric motility, likewise improvement which was expected therapeutically. So in all, there are various researches which are exploration of traditional medicine. So we will not go into depth of that. These are the researches conducted at IPGT and RA. We will escape this. So new drug development. So new drug development in contemporary context, it is a conventional drug discovery. So traditional drug research, its principles, methodologies, legislations, regulatory, there is a difference in between traditional drug discovery and new drug development. And it is a very huge difference. And comparatively as that of new drug development or repurposing, the researches investment in traditional drug discovery is comparatively very safer, very economic, and very time consuming, and there is assurance of its efficacy, as well as there is assurance of persistency in market after its launching. So if you are doing some modification, even if you are deriving new molecule, but it is, if it is from traditional drug, or processing or principle it is from traditional drug, there is more viability, more less chances of failure, less chances of withdrawal, and as it is mentioned, it is less time consuming, less economic. Likewise, in many aspects, it is there, because legislations are also, regulations are also there, very flexible for conduction of clinical trials and experimentations and researches on traditional drug research. Even other than even schedule even drugs also, for arsenicals, mercurials, and visha dravya. If you are doing some research for clinical studies, you don't need any permission for data of safety. Directly you can do clinical pharmacological experiments directly, and if there are slight alterations are also permissible. And if you are having a patent and proprietary drug, then also you don't need, if schedule E1 drugs are not there, then also you don't need any data of safety, and you can go for clinical trials. So there is huge for manufacturing also, and standardization also, there is huge flexibility. Easily you can get permissions. Likewise, and potent market, like OTC, and even more than 75% percent population of even European countries also, they consume this traditional medication. So the market is also used. So there is ample of scope in traditional drug discovery than evolving new molecules and new therapy. So scope under this drug discovery is brief introduction of regulatory aspects, ethical concerns related to traditional drug discovery, merits of research or drug discovery in traditional medicine, differences in conventional and traditional drug, challenges in traditional drug discovery, way forward to overcome the challenges in traditional drug discovery, scope and applicability or utility of conventional drug research, or latest trends in drug development from traditional drug discovery. So there are many aspects under this umbrella. We shall focus on clinical pharmacological evaluation, which is a potent tool in drug discovery in traditional medication. So clinical pharmacological evaluation, ADM studies, bioequivalence studies, evaluation of pharmacological properties, actions, studies on pharmacological properties of therapeutic aspects of drug, clinical studies like pharmacovigilance, monitoring, all those we will focus more on, and the reasons for it we shall also discuss. So drug discovery in traditional medicine is identification of possible biologically active new lead substances or compounds and formulations from traditional medicines or drugs. You can work on their extracts, you can isolate some molecules, you can isolate some, you can have some value add pharmaceutical operations, you can change the dosage form, and you can identify some parts of or fragments of that, or you can work on similar available synthetic drugs, molecules, or molecules which are there in traditional medication. So drug development using traditional drugs or medicines, formulation development of traditional medicine, that is drug and dosage form or formulation, identification of targets or mechanism of action of traditional drugs, that is, with the help of say proteomics, genomics, those could be covered under our scope or potent areas for drug discovery in traditional medication. So, what is conventional drug discovery? It is a rational drug design. Chemical modification of a known drug can also be done. Random screening. Usually, random screening is done first of all. Lead optimization is taken place with random screening is done first of all with in silico methods like structure activity relationship and say drugability and target identification, target screening. Then lead optimization is done with say in vitro in vivo research, and then it is it is forwarded to clinical research. So new indication of a known drug is a rare, but it can also be done. It is repurposing, and it can be evolved. The lead for it can be there through post-marketing surveillance or pharmacovigilance monitoring. After marketing of drug, the studies can be conducted for that. So preclinical structure in phases of new drug development, preclinical testing is there. Pharmaceutical studies are there, standardization, validation is needed first. Then animal studies like data of pharmacokinetic, pharmacodynamic, ADME studies, provisional assessment of relevant action in human. Those related studies are first of all done. The toxicity studies are done, which are usually acute chronic mutagenicity, carcinogenicity, reproductive studies, and determination of LD50, AD50, therapeutic index, minimum therapeutic dose, no observed adverse effect level. Likewise, those are needed first in conventional drug development. So, pharmaceutical research or pre-formulation studies, these are identification of potent therapeutic or bioactive or agent or molecule in silico research, that is computational methods. These are pre-formulation studies like biopharmaceutical applications, effect of say instrument, effect of some pharmaceutical operation on drug, its standards and its pharmacokinetic variants. What happens with particle size, change in particle size or shapes of particles, what happens with its bioavailability, absorption, and kinetic data. Such things are their role. Dosage form modification, route of administration changing, and targeted drug delivery, coating, encapsulation, sustained release. Likewise, so pharmaceutical standardization is a product standardization of raw material, designing of dosage form, in process standardization of finished product, and validation studies. So it may be pharmaceutical or analytical validation. The analytic, even analytical validation is also needed, because the data which is available for say contemporary molecules or isolated molecules or established procedures might not be well applicable to your formulation or product because of interference or background effects. So this is also needed. Analytical validation is also needed in context of traditional validation of traditional medication. Say clinical studies of those are classified into phase 1, 2, 3, and 4. So phase 1 studies or phase 0 studies, sometimes it is start with, there are no very firm line of delineated markings in between this. So phase 0 studies are PKPD studies, pharmacokinetic and dynetic, carried out on 10 to 15 usually volunteers, and here data of kinetic is mainly targeted. And phase 1 studies, then screening for safety, it is first in man trial, it may be first in man trial, and smaller population like 20 to 80, and drug side effects are usually recorded in this. And phase 2 studies, from this efficacy study starts usually from medium group of patients, typically 100 to 300 to determine efficacy, but it is not assured as the sample size is less. Phase 3 is a final confirmation of safety and efficacy, as well as usually multicentric and parallel trial, ranging from 1,000 to 3,000 subjects involved. And then phase 4 after launching, then after phase 3, new drug application is there, and then it can be marketed. And after marketing, then safety studies as a part of pharmacovigilance monitoring or post-marketing surveillance, then the suitability, drug-drug interactions, and specific targeted studies like specific studies in specific vulnerable groups or specific age, likewise in specific clinical conditions, may be taken over as a retrospective or prospective trials as a part of pharmacovigilance, or special studies can also be conducted in a phase 4 study. So there is a difference in between traditional drug discovery and conventional drug discovery. Traditional drug discovery is a reverse pharmacological approach, whereas conventional drug discovery is a prospective approach. Here, systematically, we used to develop a drug right from chemical identification, right from in silico lead identification, lead optimization. Every step we used to have systematic development. It is therefore it is a prospective study. And in case of therapeutic traditional medicine, the effects efficacy is already documented. Usually the safety is also already documented. It is just supported with scientific evaluation and documentation. And then those are either mechanistic studies to know the mechanism of action, or drug modification, drug development, or fractionation, or likewise these studies are there, and say narrowing the indication range or specification, narrowing the range of population, certain clinical condition or dose. Likewise, there is a value addition. Usually, there is some data regarding efficacy and safety. It is usually in the public domain. That is why it is a reverse pharmacological approach, and therefore, data of pharmacokinetic, pharmacodynamic these studies. This is lacking. Usually, the data of pharmaceutical standardization and all those, it is usually lacking in traditional drug discovery. So, but it is less time consuming, and success rate is more because it is already in the market. Withdrawal rate is very comparatively very less, and post marketing surveillance is less concerned in view of safety in case of traditional drug discovery, because therapeutic window of safety of this traditional medication is very high. If you see, yeah, develop a new molecule or a drug, it used to take at least 10 to 11 years, and it is multi-billion project. And what happens to this, if you start with say 5,000 to 10,000 molecules as a lead identification, only one will be there for approval. Out of 5,000, only one will be there. And what is the fate after this? 644 drugs were withdrawn from market in till 2014, and 344 fixed drug combinations were banned in 2016 by Ministry of Health and Family Welfare, Government of India. So usually after launching hundreds molecules in the drug, one survives after 1 year. So there is a very poor fate in after post-marketing surveillance. That is why there are countable number of conventional drugs in the market. And the Ayurvedic drugs or traditional medications are significantly safer than this, and the evidence of their efficacy and safety is far higher, because even a single expert opinion or a case report has greater value than animal study, in vitro study, in vivo research, and as those are there in public domains in centuries. It is data of meta-analysis of several clinical studies which suggest their efficacy as well as safety. So the evidence is a very much higher level of evidence is very higher, but so as to have a systematic documentation and to bring about a specificity for certain indication in a certain population at certain dose with certain mode of administration, certain adjuvant. Likewise, if you want to have a precise data and very accurate data, the further research is needed in traditional drug discovery, which can be there with just retrospective, prospective studies, as well as with pharmacological experimental pharmacology in clinical setup. Rather than adopting animal models, we can have benefits. We can directly go into clinical experimentation. Clinical pharmacological evaluations. The data of safety concern of contemporary drugs suggests that every day you will get at least 10 to 15 drug alerts in your box if you subscribe WHO drug alerts newsletter. And you will see, I forgotten to put it here, one documentation of 34 pages with more than 344 fixed drug combinations banned recently in 2021 to 2022. It is there in public domain. You can go through and you will find that many combinations are there which are banned, and you were a few years ago you were consuming them, and still those are there in the market, but those are banned. Combinations are there in the market, their prescription, concomitant prescription is there in the practice, but still those are banned. It doesn't mean that those are in the market or those were used since months ago. It doesn't mean that those are safer. So there are say signals generated for their safety concern, and that's why those are banned. And you will wonder to know even nimesulide, paracetamol combination of say antihistaminics along with anti-allergic antihistaminics along with bronchodilators. Several are there, and the market is flooded with their combinations, or it is being prescribed by physicians very commonly. So this is the fate after their even those sustained in community. Again, there is a huge concern with fixed drug combinations or concomitant administration. And your drugs, traditional medications are so vulnerable that those undergo with several processes, pharmaceutical operations, several drug combinations. You can get in a single formulation 50 or more drug, and this combination of 50 or more drug undergoes through several pharmaceutical operations like boiling, heating in various measures, soaking, fermentation, likewise. So at the end, those are safer. So there is no question that traditional medications are safer. The data of CCRAS, more than 12 publications, 12 books are there. They were in the public domain, and monographs by ITRA on Nag Basma, Ras Sindur, Ras Karpur. Likewise, we do have here more than 35 to 40 drugs, systematic development of acute toxicity, chronic toxicity, subacute toxicity we have conducted, and we have at ITRA, and we have found that more than 5 times and 10 times therapeutic dose level, those are safer. But still, there even genotoxicity study, we have conducted several drugs, more than 13 drugs. Genotoxicity studies are also conducted. Reproductive toxicity studies are also conducted. There is a systematic review article of acute toxicity studies of basmas, 11 basmas. It is also there in the public domain. And but still, their safety evaluation and documentation of their safety through clinical uses or post-marketing surveillance is needed, because the heavy metal toxicity, its counterpart, it has more wider acceptability, publicity, and strong scientific support. So it is needed that we should come up with database of their safety. And this is one challenging sector that globally, the mercurial in any form, let it be organic, inorganic, in any form, it is with the Minamata Convention, it is applicable since 2023, last year only, and now it is banned from all devices and therapies, with some few exceptions to traditional medications and some vaccine likewise. Otherwise, it is withdrawn from market. So globally, there is a consensus that in any form it is hazardous to health, biological system. It should not be there in the public domain, and therefore every attempt to demonstrate its safety is an innovation, and people are coming back to certainly they are coming back to organo-inorganic complexes and their therapeutic utilities. And it is not that contemporary scientists have not used these molecules in therapy. Injections of calcium arsenate in 1980s were there in for asthmatics and mercuries, and even this mercury chloride was very much common for therapeutic uses. Every attempt has been administered with a safety study of say uh, teen and organoteins with more than 12 models have been established, and there is a network pharmacology data with receptors, endocrines, exocrines. This slide is also not there, but there are publications that hundreds of receptors, endocrines, enzymes are interlinked to each other, and each and every linking suggests one potent research. So after even after such huge researches on arsenic stains, those are not there in the market. None of the medication is there in the market except this arsenic. For arsenic, few formulations are there, and those are indicated for management of cancer only. So the but the CDRI Lucknow has evolved with newer arsenical in the treatment of chronic myeloid leukemia. So the people are now changing their mindset, and people are wondering, but it is not that they have not tried for safer therapeutic administration, but everybody has failed. And we do have lot of information regarding their safety, vikar shanti upay, pathya apathya, concomitant administration. We have huge data of and not only the documentation, but of few formulations and identified protocols, but the principles also we do have for their administration, like Kshetrikaran, with which it is to be administered compulsorily, how it is to be administered, and how what not. So we do have this. It has immense potential for further drug development and changing therapeutic guidelines. So, as it is mentioned that there is huge difference in between modern, why it is needed to evaluate traditional medications for their safety and post-marketing surveillance and all those things, because preclinical data is not an mandatory, it is optional. So safety is optional. The documentation of safety, it is optional for traditional medications, and that is why it is needed. So we have discussed that Ayurvedic drugs are there. It is highly versatility towards multiple pharmaceutical operations, drug combinations, because these are prescribed with various anupanas, sapanas, very pathya, and in a single formulation also. Those are complex together, combined together, and it undergoes through several pharmaceutical operations. So those are safer one, and in spite of all those operations, their efficacy persists. Say mere combination of few drugs like Navaislo, Saptamrutlo, Swaskutarus, without any pharmaceutical operation, without trituration, without just mixing in traces, say 125 mg, 250 mg, those are working. Otherwise, if you administer in individual ingredient of Navaislo, say St Madhu, or in say 9 mg or 10 mg, Swaskutarus like 9 mg of Trikatu, powder of Trikatu or Pipli, likewise, it will not have any therapeutic efficacy. But when those are combined together, it has immense potential. If you see the researches on Swaskutarus, not only in vitro studies, in vivo studies, clinical studies, and several clinical studies, and all pharmacological studies are conducted by contemporary scientists, not an Ayurvedic scientist. Right? In some studies, Ayurvedic fellows were associated, but the centers were conducted, and those who have conducted were contemporary scientists. And it is well acknowledged that in combination those will work. In isolated, only 9 mg of say Shuddha Manashila will not work. 9 mg of neither Parad nor Gandhak will work. So their combination. So we do have immense potential for drug combinations, adjuvants, positive combinations in the form of adjuvants. Likewise, so we have practice of polypharmacy as well. So this is what banned drug list of banned drugs we were talking. So legislation flexibility, and there is a scope of peculiarities of Ayurvedic drugs in innovations like bioenhancer, as we have discussed, quality of life management, rasayan, rejuvenation or life prolongation, and immunomodulators. We may have in contemporary science immunostimulants and immunosuppressants like steroids, but we do have one magic tool, immunomodulators. We don't know, the same drug will act as a immunomodulator in certain conditions, in a immunostimulant, whereas it will be a immunosuppressant in some cases. The guduchi will enhance IgG, IgM under several circumstances. It will increase the threshold. So Shatavari, Guduchi has been reported to increase the threshold. Amlaki has been increased to increased threshold for adverse conditions, extreme cold, life prolongation. Likewise, those are patented along with Amlaki, along with Rasa Sindur. Likewise, but the same will be act as a immunosuppressant in case of where there is exaggerated immunity, like in psoriasis, eczema, asthma. Likewise, the same, it will not exaggerate the condition. So it is a wonder tool that immunomodulators we do have. The same drug works in both ways, but we have principles where it will be beneficial, where it will not, in which form, in which dosage form, along with which adjuvant, likewise. So there is plenty of scope for evaluation. In network pharmacology, we can have newer repurposing of the established formulations, established drugs. In maintenance of health is a complex phenomenon, and diseases are also multifactorial, and there are multiple pathological involvements, and complications of diseases or associated risks are also various. Disease is not a single entity. So the multi-component drugs are a best fit formulations in such conditions. So, but when we combine more than 3 drugs, the chances of drug-drug interactions are there. The chances of drug adverse events are increases, and if we increase drug concomitant medication more than 5, the several fold there is increase in risk of adverse drug reactions or reactions. But here the benefit, the identified formulations are there, and there are principles for their combination, for what not to combine, what to combine, where principles of antagonism is therapeutically useful, and where the combination of principle of synergism is therapeutically useful. This knowledge is there, and it can be explored with the help of current trends of in silico methods, network pharmacology, and in vivo studies, pharmacological activities. And even we can find the benefit of established animal models, that those animal models, similar animal models, can be employed directly into clinical study as an clinical pharmacological evaluation, which we'll see in few next slides. And we do have prosperity of variety of drugs. We do have Tailavargadroni, more than 60 drugs. Basic drugs. We do have more than 900 such drugs, and it is not that the world doesn't know the potential. World knows that the diamond is very precious, it is costly. Amber is a costly amber, per kg cost is in thousands of dollars, but they don't know the its utility in pharmaceutical or therapeutics. We do we have explored their utility in therapeutics. We do have only the vision that the iron can be lower. It can be therapeutically administered for variety of, rather than only iron supplementation, for variety of clinical indications, even for treatment of cancer, even for as an antipyretic, for inflammation, for obesity, for metabolic disorders, as an anti-diabetic. Under my only observation, we have submitted 2 theses on Abrak Basma, Lower Basma, where anti-diabetic activities were documented. There was Anupan of Trifla Ghana, which was subtherapeutic dose of 125 mg twice a day. That was no effect of or as a placebo. It was demonstrated in previous trials. So, but there was a significant this value addition. And for Amrutikaran also, with Amrutikaran, therapeutic efficacy increases, and the same is documented in the theses. So we do have immense potential, but there is no any clue why an iron oxide or incinerated mica will be effective in metabolic disorder or diabetes. So there is immense potential in this. For Sandhan Kalpana, biotechnologically engineer newer molecules are there, and now the world is looking forward for green synthesis and probiotics, prebiotics, and postbiotics. There is a huge chain in terms of this use of microorganisms in gut microbial flora, flora, fauna, in therapy, and none of the disease is now spared without linkage to this microbiota, microbiota. So we do have this immense potential for exploration. And in case of Bhasma, newer organometallic ligands, targeted drug delivery, newer drug delivery system. In case of Rasakalpa metal chelation, likewise, we do have immense potential. So, utilization of byproducts of pharmaceutical drug development as a part of pharmaceutical drug development, we there can be very leads in many leads in utilization of byproducts during during manufacture of Ayurvedic drugs. It will not be a part of this discussion, but it is also a potent area, like use of residue of sneha in case of avartik kshir tail, like many kshir avartik tail are there. Many indications are there. Or it could be also be a potent area for drug exploration of traditional medication, drug discovery. So there are multiple challenges, like standardization is also a huge major challenge. Challenges in uncertainty of unsuitability of certain dosage form for in vitro studies in view of their solubility. These are most of the insoluble, specifically bhasmas or rasaushadis. Likewise, there is hindrance due to partially explored multi-component formulation, because their exploration of multi-component formulation is very difficult. If we are working with single molecule, it is very much easy to have data of pharmacokinetic, pharmacodynamic, and its linkage to drug dose effect, and likewise its its passage throughout the body, elimination, everything is possible. But it is difficult when it comes to multi-component formulation. And uniqueness of principles of Ayurvedic pharmacotherapeutics and pharmaceutics, like Kshetrikaran and multiple operations, likewise Anupan, Sapan, individualized management, likewise. These are major hindrance in their exploration, but there are the ways for that also, and this can be a potent tool also. Even in vitro studies also, we can adopt the protocol of Kshetrikaran, but we should have our own tools. Even for documentation of safety also, we should have our own tools, because the documentation of safety, which data we used to submit, we used to give, it is not a sufficient data. And these are the potent tools for exploration of our effects of our drugs also. The biological half-life of these heavy metals is far higher, yeah, 3 months, years, or years together, decades together. Arsenicals remain there inside the body. Therefore, the data of 1 year chronic toxicity is not applicable as per CDCSO. This is not applicable even, or suitable to Ayurveda drugs. We should develop our own methodologies, our own protocols. This is one again argument, and very much potent tool for exploration of effect and efficacy of our drugs, that prolongation of there will be a sustained sustenance of effect for longer term. For an example, recently we have submitted one thesis on Trivanga Bhasma, where subsequently there was a improvement in glycemic control over first month, second month, and third month. And the highest glycemic control was there when the drug was withdrawn, and patients were not on any drug. But after 30 days of without any treatment, there was highest glycemic control. So there is no drug in world which will have anti-glycemic effect, anti-hyperglycemic effect after say 48 hours. So it is value addition, that drug remains for longer time inside the body. So you need to have a data of safety for longer duration, for observation, for recovery period should also be increased. Data of therapeutic toxicity evaluation should also be for longer duration, as well as it is a potent tool, as I mentioned, that the people are considering this as a top tool for exploration of therapeutic exploration of traditional medication, that these drugs have interactions with sulfhydryl groups, mercurials, arsenicals likewise, and these are very important part of many endocrines. So these drugs act on multiple endocrines. If their toxicities are there on multiple organ systems likewise, this will be a target delivery to these organs, provided that you should value add to data of safety. So the more easier it is, if as their data of toxicity, mechanism of toxicity is well established, well acknowledged. So if you data of their safety, if you are providing the data of their efficacy, is already will have greater significance, and there is no much efforts are not needed to prove the mechanism of action of these formulations, provided you should only document the safety, safety and few efficacies. That is only needed. The data of kinetic, data of all those data of mechanism of action, this is not at all needed in case of traditional drug discovery, whereas in counterpart it is most needed. So way forward is basic literary drug repurposing, utilize and pharmacological evaluation, clinical evaluation, therapeutic drug monitoring, and add-on therapy or synergistic action of traditional medication with conventional medication. But it is not possible until and unless the data of safety is established. The data of safety is more important. Safety of administration along with contemporary medications and therapeutic traditional medication is more important, because it is major hindrance. Because of this, people are not using it concomitantly. It is well acknowledged, well established, and through several studies, the traditional medications and their components or phytomolecules are well established through several clinical trials and experimental trials, that those are potent efficacious tools and potent pharmacological agents. But those are not those are less in practice because of the lack of data of drug-drug interaction studies. And therefore, if the safety in drug-drug interaction of traditional medications along with concomitant medication is established, the arena of their utilization will be increased. So there is basic difference in in terms of drug development. Yeah, the more important is the data of therapeutic drug monitoring. Therapeutic drug monitoring is a potent tool in traditional drug discovery, because it has high therapeutic high evidence of level of evidence. And why it is more needed rather than having clinical trials, the therapeutic drug monitoring of traditional medication is a very potent tool, and it should be utilized, because here there is you can have data of millions of patients in therapeutic drug monitoring, vice versa in thousands or hundreds in clinical trial. Duration can be years or decades as a prospective retrospective trial. At a single time you can have a data of 10 years ago. Also you can take out from therapeutic drug monitoring. Concomitant medication illnesses usually present, these are present in therapeutic drug monitoring, which are avoided in clinical studies, and dose is variable in therapeutic drug monitoring. Conditions are flexible, therefore more information is captured from therapeutic drug monitoring, which is lacking in clinical trials. So we should have our own sectors, areas of priority areas of traditional drug discovery focus. That therapeutic drug monitoring is not more important and more important tool in drug discovery in of concomitant contemporary medications, but it is more important tool in case of therapeutic traditional medications. So why specific therapeutic recommendations are mandatory for therapeutic drug monitoring? Usually we used to do that. We should have data of Agni, Koshta, Prakriti, Bal, and sleep pattern, and concomitant administration. All those are needed and if those are judiciously used, then only the efficacy will be assured, or it will be have more therapeutic efficacy likewise. But are these not needed for contemporary medications when those are developed? Whether paracetamol is works same in pitta prakriti, kapha prakriti and all, and whether it is only applicable to say Ayurveda drugs? Yes. So for Ayurveda drugs, it is well acknowledged, well needed. Otherwise, the data of therapeutic utilization of drug in all those conditions is very important, and the researches on this aspect are very much needed in case of traditional medications or Ayurveda medications, whereas those might not be needed in case of contemporary medications, because there is basic difference in modern many contemporary medications which are administered enterally or orally, and traditional medications. Usually for traditional medications are not drugs. Most of the times those are prodrugs. The what you administer doesn't work. There is secondary metabolites, and it's metabolites, secondary tertiary metabolites usually used to work. For an example, if I talk about fatty oils or snehakalpa oils or ghee, without gut digestion, Kshirpak, without gut digestion, those will not be there in systemic circulation. So there will be first drug modification, metabolism in gut, second metabolism in say liver, or in, through lymphatics, those will be going to systemic circulation. So that nature of drug varies. So there is a basic difference that contemporary drugs are made 100% water soluble, so as to have 100% bioavailability, whereas you cannot expect 100% bioavailability and 100% solubility in case of traditional medications and drugs. So you need to you expect the action of drug, sustained action of drug, you expect the action of drug till large intestine, till colon, rasaushadhi till colon. So drug interaction, what you have administered, co-administered along with what diet you are taking, their drug interactions and their secondary tertiary metabolites and their post metabolic interactions, pre metabolic interactions are part of therapeutic Ayurvedic or traditional therapeutics. So we cannot expect, and the researches are needed in this context, and there are potent researches that fatty acid complexes of iron are patented in 2002 for their sustained release and more bioavailability, which are, as we discussed, which is nothing but a similar process as that of Amrutikaran, and we have got more results when Amrutikaran was applied to Abrak Bhasma. The more therapeutic benefit in case of diabetes was there. So the similar observations we used to give. That is why these principles are and researches in these aspects are needed in case of traditional medication. We cannot avoid them. And in clinical studies, it is not possible that we can work with all prakriti, dose, duration, and all in all clinical variants and variants of pharmaceutical operation and variants of formulation in all possible variants. One cannot have data of in vivo clinical studies. So therapeutic drug monitoring or data of pharmacovigilance in case of traditional drug discovery. So why this is a traditional therapeutic drug monitoring is needed? It leads to drug withdrawal, modify labeling, safeguard to promote safe use of drug, restrict indications, additional contraindication. It adds additional precautions or monitoring or warning, recognition and quantification of adverse effects. And to this, it value add to therapeutic recommendation, generation of database for therapeutic effective dose, and with which combination, with which dose, in which prakriti, if it is given, it is more beneficial than others. Likewise, generation of database for clinical safety evaluation of Ayurveda days to update the therapeutic recommendations like alerts, etc., and generation of database of possible new indications, that is repurposing. With monitoring, you can know that in a certain dosage form modification or certain dose is having, you can get newer benefits in the same patient. The drug may be prescribed for some other reason, but you may get some other benefits in some other conditions. So there may be repurposing of the drug with therapeutic drug monitoring. So the therapeutic drug monitoring could be non-targeted, or it could be targeted monitoring, like target drug distribution, consumption surveys, drugs of various categories in Indian market, and in specific conditions for Schedule 1 drugs, or drugs in critical disease conditions like liver failure, alcoholism, Rupka Rog, Shodh, Gulma, Hridrog, likewise, and clinical vulnerable patient like pediatric, pregnancy, lactation, geriatrics, likewise. Their safety can be documented in G6PD deficiency, liver impairments. For all those conditions, one cannot you cannot have clinical ethical permission. So therapeutic drug monitoring, if you want to know the efficacy, safety, and what should be the more therapeutic benefit in which condition, along with which anupam, with drug combination, likewise, if you want to know the data, that will be there through therapeutic drug monitoring. So that is why it is needed. And in sensitive cases, emergency medications also, we can have the ethical permissions will not be there, so that therapeutic monitoring is an ultimate option. So potentials for therapeutic drug monitoring, why it is needed. It optimum utilization of existing clinical health infrastructure can be there. Whatever it is being done, we can use those resources. We don't need additional in either recruitment of manpower or drug or people for monitoring and all. It is utilization of existing infrastructure, resources, instrument, investigations, practices, all those things. With the help of this only, the monitoring is needed and documentation is needed. Clinical safety documentation is done, clinical precise medication is done, clinical safer medication is done, and medical research. It is optimization of therapeutic recommendations and optimization of clinical or therapeutic outcomes, and removing gap of translation of in vitro researches on safety efficacy directly into clinical practice. There are clinical researches of safety in vivo studies are also there on toxicity study, but with this therapeutic documentation, we establish that those are there are no gaps in their application in therapeutics, and those are safer in clinical conditions also. So then there are various sources of data collection. We shall escape in this. We are running out of time. Within next 5 minutes, I shall conclude. So there could be drug-drug interactions, bioequivalence studies, single dose studies could be there, which is the document. The heading is we lost the heading. It is experimental evaluation, experimental pharmacological evaluation, clinical experimental evaluation. So you can directly go into clinics rather than having those the data of drug responsive drug and dose, drug response and dose. You can have in clinical study it is not possible that you will work with different doses and same drug, and so you can have several clinical trials. It is not possible there. So you can work in if you are working with single dose like intestinal motility, anti-hyperglycemic effect, bronchodilation in asthmatic, diuretics, analgesic effect, cholinergic, antimuscarinic. With single dose, you can document the effect. So it is a job of clinical pharmacological evaluation. There are and similar protocols which are there in vivo. Some protocols that can be adopted directly into clinical practice in case of traditional drug discovery. So drug food interactions can be there. Safety of drug, comparative efficacy studies, additive, synergistic effect, antagonistic effect. If the value added, if on ongoing anti-diabetic drug, along with that, if value added traditional medication is added, whether there is safety, whether there is hypoglycemia, whether there is a more improvement in glycemic control, whether there is comparatively more improvement in, say, reversal of complications of diabetes. Likewise, there could be many purposes, and only we have to monitor. We don't have to administer medication. We don't have to recruit manpower or patients for this. So changes in pharmacokinetic, bioavailability, excretion can also be there with single drug or drug-drug interaction, drug-food interaction. Likewise, interdisciplinary safety may be established. The major drawback, as I mentioned, that why people don't use to have Nisha Amlaki or Amlaki or Haridra along with say metformin, because some data, some studies demonstrate that there is a less bioavailability, like work with Nisha Amlaki and their reduction in area undercore of metformin around 15 to 20% of reduction in bioavailability. But the another research, in vivo research by Bhushan Patwardhan sir et al., it suggests that in spite of this reduced metformin level, there is improvement in glycemic control, improvement in lipid profile. Likewise, so such data are needed. Until and unless those are evaluated, until and unless researches are done, their optimum utilization as a adjuvant medication will not be there. So this can be done in a single dose over duration, and well-established models, in vivo models, can also be adopted, as well as in clinical pharmacological evaluation can also be done with this. So even with therapeutic administration, traditional medications, we can go for data of metabolite analysis in urine, what happens with metabolites in serum, like metabolomic studies, like may be a potent sources for potent metabolites, which may with the concentration of metabolite, then there it could be a lead of uh, such metabolite could be there by fermentation, by some hydrolytic processes or oxidation, or some pharmaceutical modifications. Such metabolites may be increased as a pre-treatment, and which may be taken from Ayurvedic pharmaceutical operations only. We may choose that which form this dosage form is more helpful. Whether the duration of immersion is, if you improve, the metabolic concentration is increased and the pharmacological activity increase. So it could be a lead optimization with the help of metabolite evaluation in blood, serum, or biological fluids or urine. Likewise, even in saliva also, even in CSF, we can work directly, because there is no need to have data of safety and subsequent all phases of drug development. You can directly work clinically and have value addition exploration of mechanism, and new lead molecules can be there from traditional medication. So so there could be two period administration. One drug may be administered say morning, and another at evening, and drug interaction could be there without interaction in gut. Likewise, two sequence can be altered. First drug is given prior, and another later on. The mode of route of administration may be changed. One may be injectable and others oral. So post metabolic interactions could be evaluated. Likewise, there is huge scope, and medications which interfere with anticoagulants, like increase INR, PTT, like Lashun. Many anticoagulants are anticoagulant properties of many medications are already documented, but their formulations and their useful therapeutic utility can be further improved with the, and it is very simple one. It may be there in very simpler models are there in, it may be there in next slides, that you can have with simple weight of clot. If you take out the blood with administration of your drug along with standard control, without standard control, placebo, you can check with thrombolytic property, anticoagulant, just with weighting the measuring the weight of clot. Likewise, there are very simplest models, and those are published in very reputed journals like Journal of Angiogenesis. Likewise, so with simpler modalities, there could be therapeutic exploration. So I am committed to complete it in 5 minutes. So we shall
Yes,
move
yes,
toward
please,
to
please.
recent trends in drug development. So there could be a concomitant medication with contemporary medications. There are a lot of possibilities of working with this. So So clinical pharmacological evaluation is a huge stream. It includes drug utilization studies, pharmacoepidemiological studies, pharmacoeconomics, emerging role of clinical pharmacology in biologics and biosimilars. Likewise, many applications it do have. So we shall just this the superiority of reverse pharmacological approach is, as I mentioned, that huge database is there, less economic involved, less time is consumed, more safety is there, and clinical applicability is huge, market sustenance is assured, and technicians expertise and manpower, which is we don't need any specific technical advancements for its application, and high instrumentation technology we can use, but it is not needed always that instrumentation, high end technology, sophisticated technology is to be always used. So with merits of clinical pharmacological evaluation, we can find the benefits of in vivo, that is animal studies experimentation, that is cohort is there. You can control the environment, you can control the conditions. These benefits you can take if we work with clinical pharmacological evaluation directly clinically. So so we can work with 24-hour urine excretion, likewise. So we can work with our drugs. We can explore our action of our drug with mechanism of action of studies. That can also be done where your drug is having interference with P cytochrome 450 enzyme, that is a major metabolite, drug metabolizing hormone, which suggests that this drug will have some drug-drug interaction. It could be a positive interaction, negative interaction, but usually if it is inhibit this P cytochrome 450, usually the co-administered drug, it will have more biological half-life, and in less concentration it will be working. There your drug may be metabolite enhancer, bioenhancer like alcohol and pesticides. Likewise, it may be p-cytochrome metabolite inhibitors. So these activities can be enhanced, and from that you can know what the positive drug-drug interactions. Without actual conducting a study, you can conclude what will be the possible drug-drug interactions with which drugs, with which contemporary drug or your drug. And there you can even establish whether your drug have chronotropic effect, inotropic effect, dromotropic effect, adrenergic effect, cholinergic effect, insulinogogue. Likewise, with just a single drug administration, single dose administration, few administration, you can check the levels of what happens with heart rate, rhythm, levels of corticosteroids. If levels of corticosteroids changes, that means the levels of insulin, levels of its analog hormones like adrenergic will be changing. Those are all interlinked. So with this evaluation, it has multiple implications that you can directly clinically you can work with your formulations and document it. So it is a potent source of exploration. Biological markers, we can you have mechanism of action. We can know, as I mentioned, that metabolomics. We can know the proteomics, lipidomics. By documentation on proteomics and lipidomics, we can document on safety. If proteomics is not changing, the drug is safer. And with the most wonderful thing with working and exploration with traditional medication is that there are very few sectors where even failure is there, negative results are there, you have positive gains. Whatever the results are there, the negative results, positive results, or none results, in all cases you have it's huge implications. If there is a change in proteomics, your drug may be working. It may have several implications. If in case of disease, if there may be reversal of some pathogenic proteins, you can document. If there is no change in proteomics, your drug is safety of your drug is established. In any way, you are getting benefited with these explorations. So gene expressions, mRNA expression, likewise we can work with PCR techniques likewise. And measurement of endothelial function, few flow measurement we can do with cell adhesion, platelet adhesion. There are confocal microscopic and many models we can adopt in live. Those can be there. Pharmacodynamics of proteomics, RBC fragility assay, metabolomic assays with NMR, MS, likewise, those can be done. Immunomonitoring and evaluation of function of components of immune system, IgG, IgM, and specific immunity against specific challenges, specific vaccines, specific diseases, specific antigens can be documented. Then pulmonary function testing, pulmonary challenge models, neurophysiological and cognitive measurements, assessment of brain function by MRI, PET, polysomnography, measurement of renal function, inflammatory ex vivo and in vivo models in man. There are several inflammatory models. You can evaluate and inflammatory action of your drug in clinical settings. What we used to do in say immunity models on immunity, acquired immunity in COVID, IgGM, acute infections, IgG, long-term infection, specific antibody titers on several diseases, that can be with co-administration of your drug, prior administration of your drug at different doses. We can work with this. So metabolomic studies, as we will give to lead optimization. Then enzyme inhibition can also be there, like say enzyme inhibition. So alpha amidase, alpha glucosidase, likewise, whether it is so by that we can know the mechanism of action of our drug. And those several enzyme inhibition studies are there. And several enzymes in tissue culture, we can know the whether it is stimulatory to some receptors, some organs, some endocrines, exocrines, or inhibitory. It does the down regulation or up regulation. Their evaluation techniques are there. So hepatic enzymes and endocrines we can use with the help of this hepatotoxicity, cell toxicity, or its upregulation, down regulation can be there with the help of bilirubins and their SGOT, SGPT documentation in cell line studies. Likewise, there could be many implications. The use of cell line studies can be applied. And even from in vivo study, from clinical studies, the cells, in vivo cells can be cultured, particular cells after drug treatment. Cells can be there, in pathological conditions, cell can be grown, and on these pathological cells which are having different mRNA expression, the change in mRNA expression after drug administration can be before treatment, after treatment. Likewise, we can document even gene expressions, likewise inhibition it could be reverse, as a reverse pharmacology. The same genes, proteins, cells may be taken, injured cells may be taken, pathological cells may be cultured, and on these cultured cells the drug may be in vitro, tested in vitro, or that with therapeutic administration the reverse changes may be documented. So the utility of in vitro say may be documented. As we may mention that hemolytic activity that can be documented. There are publications in Journal of Angiogenesis. There, as we discussed, that models of inflammation, UVB irradiation, histamine provocation, KLH challenge test, several models are there which can be adopted to document anti-inflammatory effects. So we shall go to actually the main exploration, is a therapeutic exploration of traditional medication in drug discovery. So these are the models in sensitivity test, likewise. But we shall, as time doesn't permit, we shall go for contemporary drug development. So there are neurophysiological investigations can also be there, and you can document whether it is CNS, your drug is CNS suppressant. Likewise, what happens with alpha waves, beta waves with polysomnography. Likewise, there are multiple models, and you can document electrodiagnostic techniques are there, EEG, yoke potential, ANS disorders. For ANS disorders, you can do physiological testing like orthostatic blood pressure, head of tilt test. What happens with these conditions, and how whether your drug will be useful in ANS disorders, whether it will suppress or augment the activities of ANS, CNS. Likewise, whether it will be sympathomimetics, sympatholytic, likewise. Specific autonomic test can also be there, sweat test, Valsalva ratio. Likewise, before administration of drug with different dose variations, anupan, this all these experiments can be done. Even electromyography, electrical impedance micrography, all those can be conducted. The Nimhans, Bangalore, so several neurology institutes, these tests are commonly available. So endocrinal evaluations could also be there. Evaluation on cardiovascular effects, anticoagulant, antiplatelet, with respiratory plectrophometry, renal systems also. Even the basis of classification of drugs as volume, say loop diuretic and all, it is just dependent on urine volume, electrolyte excretion at timely intervals. So all this can be clinically done. So anti-asthmatic effect, histamine test, likewise. Lactation studies could also be there, whether drug is galactogog, and if the drug is galactogog means it is working on dopamine. Likewise, there are multiple implications of this indication. So latest trends in drug discovery is first of all in silico applications, then as a lead identification, then lead optimization is done with in vitro studies and in vivo studies, then in silico methods for drug screening or virtual screening, that is bioinformatic tools. With this we can whatever the drug we have designed, we can check its drug likeliness, the bioavailability, probable bioavailability on the basis of in silico analysis, computational methods. There could be a drug likeliness with several rules are there, Lipinski's, Weber's, Gore's, likewise, and there are several websites, online websites with which it can be done, like Swiss ADMA analysis, Swiss target. You can have target prediction. You can have data of absorption, distribution, metabolism, excretion. Molecular docking can also be done. Structure activity relationship of certain molecules, extracts, or after say fermentation, after some pharmaceutical change, we can document change in structure activity relationship, change in network pharmacology, change in targets can be documented, and the changes in chemical configuration will be documented with the help of LCMS analysis. Likewise, many evaluation tools, and there are this online available websites from which it can be done. Several websites and several tools are there where you can even generate the structure, you can change the molecule, you can change the you can generate the molecule, you can test the target with target, its affinity can be tested, and you can do alterations, and you can know the by which pharmaceutical operation this alteration will be there, and what will be the change in its target delivery. So all these can be incorporated there. In in vitro studies, in cell line studies also, bioavailability, pharmacokinetic, pharmacodynamic, toxicity, toxicokinetic data can be generated, and this can be equally applicable to traditional drug discovery, but there are some limitations in application of this data to clinics. So protein identification can also be done with PCR, ELISA, whole targeted genome sequencing, like 16 mRNA, 8 mRNA analysis, DNA, RNA with GCMS, LCMS analysis, urinary metabolites can also be there. So for systematic drug development, everybody knows Schedule Y of D&C Act is there. I will not go into depth of this. So with this, I think we should stop. There are multiple ways. Say as I talked about for in vitro models, mechanism of action, anti-diabetic activity, inhibition activities. For an example, this slide is there. 5 mechanistic studies for inhibition in enzyme inhibition are there, and for all diseases such inhibition studies are there, and with just spectrophotometry it can be done. It can be done in vitro studies, it can be done in pharmacological clinical studies. You can take a blood sample and your serum can be tested for say antioxidant activities, for glucose uptake, for inhibitory alpha amylase activity. Likewise, even serum, treated serum with drug, without drug, likewise those such assays can be done. You can administer serum and urine to test whether what happens to hepatocytes, whether it increases SGPT, bilirubin, likewise whether those are it down regulates, upregulates. Even you can administer them in while cell culture, and you can know the differences. And the significance is that there is no gap in there is no gap in application of this in vivo in vitro results in clinically, because you are working clinically. You can directly take treated serum, treated biological fluids, and check for in vitro analysis. So these are the potent methods which can be transformed for exploration of traditional medications for their lead identification. So in vitro interactions like chemical arrays, we do have right now chemical protein interactions, enzyme activity, inhibition, target identification, pathways, cell-based functional analysis, and chip-based arrays. Now we do have all chips for albumin, microalbumin, glucose. We do have kits, and all kits are available. So we can make use of this. There is cassette dosing. Multiple drugs are given simultaneously, and safety or efficacy is evaluated. So you need less number of patients likewise in this. So there are many benefits in this. We shall not go into this. There are fluorescence techniques are also. We shall escape this, and flow cytometry is also there, as I mentioned that for thrombolytic and inflammatory endovascular diseases and vascular diseases. This is blood flow and flow cytometry. This is a very important tool. Live flow studies are done, and even with flow cytometry, the count of cells that can also be done. CD3, CD4, CD8. Likewise, with drug, this can be evaluated. So chicken embryo models, small in vivo models are also there for angiogenesis. Likewise, so use of microbes in pharmacokinetic pharmacological evaluation, even candida albicans for pathogenicity mechanism, and their metabolites can also be tested. So upregulation, down regulation of enzymes, as I mentioned, in vitro or in biologicals or in organisms can also be tested. So omics studies are also tested in vitro cell lines. So with this, I would like to conclude. I'm very much thankful to organizers, Institute of Statistical Applications, CCRAS, as well as, and to the director ITRA for giving me a chance to express me. I'm very much regretful for not being confined to time limit and the lacuna in sharing this proper PPT in a mode. Thank you. Thanks a lot once again.
Thank you so much, sir, for delivering a wonderful session in this 3-month certificate program. Now, all delegates, if there is any query, you can send us. Due to shortage of some time, we have another session. So if you have any query, you can send us. I will forward that queries to sir, and we'll revert you as soon as possible. So thank you on the behalf of organizer. I convey our sincere thanks to Dr. Prashant sir for delivering a wonderful session and sharing expertise with all delegates presenting in today's online session. Thank you very much, sir, for your valuable time and expertise. Thank you, sir.
Thank you. Thanks a lot.
So all delegates, this is the end of today's session. Next session is scheduled on 3rd June, moving towards stats and biostatistics, that is selection of research problem and formulation of hypothesis. Tomorrow there is a assessment 1, that is MCQ type. We'll share the link in the WhatsApp group. You could go through it, and till 3 June you have time, so you can revise all the questions we just delivered. So this is the end of today's session. Thank you everyone for joining. Thank you.